Welcome to our webinar titled, What is Chemo Brain? Diagnosis and Treatment. Thank you for joining us. My name is Lee Clark, patient educator, and I'll be moderating the presentation today. As we get started, I'd like to recognize the generous support of our corporate sponsors and the generous support of our patients, family, and caregivers for supporting the webinar today. Due to the high volume of teleconferences on the internet, it is possible you may lose your connection during the program. If you are unable to view the webinar online, you can call in to hear the audio portion of the program using the call-in number in your reminder email. Today's program will be archived to our website within 2 to 3 business days. You will be notified by email when it is live and ready for viewing. Immediately following the presentation, there will be a question and answer session. Please feel free to submit your questions at any time during the presentation. To submit a question or comment, open the Q&A window in the lower part of your screen. Type your question or comment in the small text box window, and when you have finished typing your question or comment, just hit enter. We will do our best to get to all questions today. When asking questions, I do ask that you do 2 things to help me manage the incoming questions. First, submit your entire question all at the same time. Second, please do not share private health information in your question. Our speaker cannot answer any specific questions related to your health care. Today's specialist is Dr. Jorg Dietrich. He is the director of the Cancer and Neurotoxicity Clinic and Brain Repair Research Program at Mass General Hospital Cancer Center and is an Associate Professor of Neurology at Harvard Medical School. With that said, it is my pleasure to welcome Dr. Dietrich. Thank you for joining us today.
Thank you very much. It's my tremendous honor and pleasure to join this webinar and to be invited to present on this important topic that is relevant for many of you in the audience, and obviously for me as an oncologist treating patients for cancer. And with that said, I would go ahead, I think, and share my screen so that you will be able to see my presentation. Okay, so first my disclosures, none of which are relevant for today's presentation. And my presentation or my talk is divided in 3 major sections. The first one is to outline really the clinical problem, is what some of you may have experienced throughout treatment or after treatment. What is chemo brain? How is it defined? What are the symptoms? And what are the issues that are encountered in clinical practice? Second, what is the current understanding and the underlying mechanisms of why we think there is such an entity of chemo brain, or in other words, a condition that affects our mental capacities and cognitive function? And lastly, what can we do about it? How has research informed current patient management, and what are existing and hopefully future options to improve quality of life in affected individuals? So first, to the clinical problem. This is something that you've heard or personally experienced in many forms and variations. And one is the memory loss. It's one of the most common symptoms that are notable in patients, both subjectively and objectively. And that can come in very different forms. So patients might complain of issues with, I just forgot the names. I have difficulties coming up with the right words. Or we had a conversation earlier this morning over breakfast. I really don't remember anything what we talked about. Or you misplace items and things you don't find as well around. You sometimes perhaps a little bit disoriented about things. And that can come and go. And there might be days and hours where everything is perfect, and then days and times of the day where things are just not as good. And so we will go over and dissect a little bit what the issues are and what we have learned over the past, perhaps 2 decades. The first slide here really summarizes that this is now a phenomenon that has been pretty much established in the scientific world and is not shoved aside as an issue of, oh, this is just a cancer patient who has trouble with mood, they're going through a hard time, and this is all that there is. But chemo brain, or the impact of treatment on brain function, has been acknowledged as a real entity that can be studied, and that has been acknowledged and accepted by many, not all, but many oncologists as well, and are now being discussed, for instance, even prior to treatment initiation, that cognitive function might be one of the potential unwanted adverse events. This cartoon gives you a summary of what are cognitive domains that are relevant. One is attention and concentration, that locate to the frontal cortex or frontal lobe. And they travel along with issues like processing speed, how quick you think about a certain concept, executive function, how you can complete a task, have an initiation to do a task and start it to also complete it. Reaction time is something that can be easily measured. And then along with regions in the so-called temporal lobe of the brain, issues that are affecting memory function. Usually it's short-term memory function, and that obviously has impact on how we learn and how we can use information that is stored, how this information can be retrieved, and how we can use our memory or not in case we are affected. So the interesting part is that the prevalence of any of these domains being affected is fairly large, up to 80% in individuals, but not everybody is affected. And this is still one of the biggest issues in the field, is what are the key risk factors, who might be affected and who is not, and how can we influence treatment choice and modalities according to that. One thing that I wanted to point out is when it comes to chemo brain, and we are all too quick in attributing the chemotherapy drugs that have caused this, that there are many other factors that can significantly influence our cognitive function. And among these are certainly genetic factors. I've had many patients who received the exact same treatment, are at the same age group with the same disease, and one might be really suffering from side effects, cognitive side effects, whereas the other has no issues whatsoever, is fully working and maintaining their high level of performance and functioning. But issues such as fatigue, they're ranking almost as high as cognitive and memory issues. Certainly mood disorders can contribute. Sleep disorders, which is not on this slide, but can have an impact on cognitive performance. And then some of the features that have been studied, such as endocrine therapy, oxidative stress on a cellular level, mitochondrial damage that plays into this, and then even the effects of cancer itself have been quite astonishing as research is being performed and studies come out that many patients, even because of the tumor itself, could have, even prior to treatment, impairment in their cognitive function. So when I, as a physician, see patients, we have certain tools in place, and one of them is imaging. It's imaging of the brain, typically done with an MRI scan, magnetic resonance imaging. And in the worst case scenario, one might find abnormalities, as you can see here, in 3 individuals of patients that I have been involved in treating. On the left, someone who received high dose of methotrexate for an underlying lymphoma. In the middle, a dark combination of both methotrexate and cytosine arabinoside called ARA-C. And on the right, someone with acute myeloid leukemia who did receive intrathecal treatment, so directly into the cerebrospinal fluid space. And in the right upper corner is an MRI that is from a normal individual. So what you really appreciate is the vast, extensive white matter injury, which is sort of everything on the subcortical level. So we have the cortex that harbors all the neurons, and subcortical is where the myelin, which is the insulating material around nerve endings, is located. An injury to that area can have tremendous effects on how our brain works or not. And here's a cartoon of how this myelin substance looks like, how it ensheathes these axons, and is really relevant for an appropriate neural function. You can study it in electron microscopy on the lower right with thick layers of lipid and protein. You can also study it in culture and see how elegant these cells and how complex and complicated they are. And these cells can be at risk for damage in the context of treatment for cancer. And so here's a cartoon from one of my earlier collaborators of why is this relevant? Because the myelin really allows signal conduction to happen along an axon, along a nerve cell. And if the myelin falls apart, this signal can't travel anymore. And so the nerve function really subsides and ends. And with that, you have trouble by, for instance, thinking and with memory function. So mechanisms have been studied based on preclinical work in cell culture, in animal models, but also in patients. And that will now be the second part of my talk, to really dive into what are the issues and perhaps why are some patients at higher risk than others. It all started off with essentially Ramon Cajal, who was a Spanish Nobel Prize winner for his seminal work on brain architecture, with very elegant identification of what cell types exist. But he set forth a dogma that took almost a century to be thrown overboard. And this dogma is what you can see on the lower part, is that essentially what all we have in our brain is the function we are born with. And there's not much remodeling throughout adult life, even throughout adolescence. And essentially cells only have one choice, it's either survive or die. But he ruled out the possibility that new cells are born, new nerve cells and new support cells throughout life that could maintain neurological function. You may wonder, why is he showing us a list of songbirds? And I think the songbirds have in the understanding of brain plasticity a very special role. It was the songbird really who allowed in its study to identify that new neurons, new nerve cells can actually be created and born in adulthood throughout life. And this was found because songbirds can learn new tunes each season. They can modify ways how they acquire new melodies and tune, and the songbird behavior is influenced by this. And so with that, a lot of investigators and neuroscientists asked the question, if a songbird can do that, how is it in the mammalian system, in rodents and eventually in humans? And it was found that there are two major regions in the brain. And this is a rodent brain, not that you need to remember this, but there's one region, it's called the subventricular zone, SVC. And there's the other region called the dentate gyrus, DG. And both of these regions are critically relevant for maintaining brain plasticity throughout life. So new nerve cells are born in these regions, and in the rodent, they migrate along a pathway to the olfactory system, because the rodent is very much obviously in tune and dependent on an olfactory system to navigate and to survive. And this system is relevant for humans too, but not as critically relevant. We might survive still if we don't smell well, and you have had this now with COVID-19 as one of the symptoms, that a sensation of smell, the olfactory system, is targeted. And then lastly, the dentate gyrus, which is this region here amplified, an area that is in the temporal lobes, so brain areas behind your eyes, and that is critically relevant for maintaining the functions of memory and learning. And so if you knock out these two areas in the brain, animals as well as humans have a hard time of maintaining their function. This is obviously much more studied in cell culture and in preclinical model systems, but we know from a lot of indirect evidence from humans and patients that these systems still play a role as well in our very complicated organism. And here are a few examples. This was a fantastic study done a long time, 20 years ago, that identified that this hippocampus, this area I just told you about it, which is so relevant for maintaining memory function, is much larger in individuals who use a high level of cognitive function related to this domain in the brain by using navigational skills that are relevant to get through a large city like London. And so they studied the ability to perform, and this was in the area without GPS systems and without a lot of smartphones that we have nowadays to compensate and actually take away some of that skill from us. But the bottom line is, the more you use your navigational skills, the more this area of the brain is actually developing, which is the hippocampal system, right here in this cartoon, highlighted in dark blue. Another one is an intriguing study that was done in students and published in Nature about 15 years ago, and which is, if you take up a new skill, such as joggling, it's not something we do on a routine basis, that you can actually influence brain architecture and structure. And that is a fascinating concept where through learning new skills, you have the ability to actually change how your brain functions. That's something that is now being incorporated in sophisticated rehabilitation efforts, especially after, for instance, wartime injury, veterans that come back and need to be trained. So by using knowledge like this has helped a lot to think in a whole new, different way about how the brain evolves, how the brain maintains its function, and how can we help to repair damage. One thing that is very, very important for you to remember when I give the rest of my talk is that certain cell types in the brain, and here you hear a few names, astrocytes is one of them, the neurons you've heard about, and the oligodendrocytes are the ones that make that myelin substance. They all need to act in concert. And if one is not happy, then the others will suffer. It's like a healthy family that works together, or a healthy forest. And if you knock out a few elements, then many other elements of that biotope will suffer. And the similar concept really applies to the brain, that you can occur damage and as an end result, lack of function through interference with many of these cell types. This is a cartoon as well that gives you a flavor of how many cells are necessary to maintain that stem cell niche where new neurons and glia can be born. And so I'll just give you again the word of an astrocyte, a star-formed cell. There are these precursors that form new neurons, but there are also the blood vessels, small capillaries that really form a niche in which other cells cluster around. And if you imagine that you would damage blood vessels, you will damage a lot of that unit in order to maintain its function to form new neurons throughout life. And here's an example from some of our own work where you can study these early cells labeled in green. These are early myelin-forming cells, and as they mature over time, they form longer processes, many more processes to begin with, and take on different properties. And eventually, with this labeling system of GAL-C, which is a surface marker, you can identify them as mature oligodendrocytes. And at that stage, they don't divide anymore. They don't keep on making new cells. What is really relevant is the early precursors or progenitor cells that are relevant to maintaining that there are enough of these myelin cells later on. So if you look now in animal model systems exposed to chemotherapy drugs such as 5-fluorouracil or Ara-C, you see that one of these structures, which is here, this band, which is called the corpus callosum, is rapidly thinning out over time. And so they lose this myelin over time after repetitive exposure to chemotherapy. And you can study this elegantly in electron microscopy as well, where these myelin sheets really thin out. And here you get an appreciation of how dense in the normal condition this particular brain structure is, and after injury following repeated exposure to chemotherapy. And then many other cells can be labeled, and this is what the advantages of neuroscience systems, of microscopy, of surface markers. We can really dissect many of these cell types to understand which ones are suffering more than others. So this just gives you a flavor of how much this can be understood on a cellular level and even on a molecular level. Now, jumping back again to patients, we asked when patients that I treat many times have brain tumors or brain cancer, what is the impact of the treatment we offer to our patients to keep them alive, to get rid of the tumor or stabilize the disease? What is the impact on normal brain? And you see some striking changes before exposure to chemotherapy and radiation and after. And this is just after about 35 weeks. The brain itself loses volume. These fluid-filled spaces in black get larger over time, and we can now even assess with volumetric analysis whether the gray substance or the white matter suffers, and it turns out that both over time do suffer. So in short, we know from studying patients prospectively and longitudinally over time that there is quite a bit of a change in terms of structural differences that happen over time. What about brain function? So glucose, which is a sugar molecule that we have in carbohydrates and even fats and proteins, will eventually be metabolized to form sugars. These sugars are necessary for us to perform. So if I give my talk now, I really need glucose to help me think through the slides, and you will do the same. But glucose metabolism can be measured with a modality called PET scan. And so whatever is red here is a high uptake of glucose in a normal or pre-treatment individual. And after exposure to chemotherapy, you can see that glucose metabolism goes down. And we don't know why that is, but it's a striking finding. And this was a study that we did in patients for lung cancer. None of these patients had any issues in the brain. They had no neurological condition, no psychiatric condition. We just wanted to find out whether a chemo drug used to treat lung cancer can interfere with brain metabolism as measured by glucose uptake, and it does. And so this was a striking finding to people who use this modality to measure where tumors show up. And probably some of you have had a PET scan, and that really loses the utility of glucose. I give you one other important finding and concept, which is cognitive dysfunction or cognitive function per se depends on so many other factors. And I mentioned this earlier in my cartoon slide, but stress is certainly rising your steroid levels. Sleep deprivation does the same thing. Behavioral changes and mood-related issues can interfere with cognitive function. And this concept has been so well studied, in particular in the context of forming new nerve cells. So if, in very simple terms, you are very stressed out, you don't sleep well, and you are depressed from whatever reason, and now on top of that, you're exposed to chemotherapy, you have a much higher chance of having a decrease in the ability to form new nerve cells throughout the rest of your life. And so this is a model that I show often to my scientific colleagues, but bear with me, it sounds perhaps like a different language to you, but what this really depicts is that on very different levels, in lighter colors going to more deeper colors, there couldn't be injury could happen on the level of direct cellular toxicity, such as myelin forming cells could be at harm after certain drugs. But you could have also developed chronic inflammation that doesn't really go well with brain function. There could be the issue of oxidative stress. Too many free radicals are forming in the context of cancer and also exposure to chemo drugs. And that in turn leads to activation of a lot of police cells, like the microglia, that cause additional damage sometimes. You could have impact on the mitochondria, which is sort of the basic factory in each individual cell. And that can cause loss of the ability to form new neurons throughout life, vascular damage on your blood vessels, myelin damage on insulating material, with the end result of what we call neurodegeneration. The brain is not working as well, and certain elements are suffering. And so, not surprisingly, scientists, including myself, have worked on a concept that might explain some of these features that we see in terms of cognitive dysfunction or impairment, which is that cancer patients per se might have a different trajectory in terms of aging when compared to non-cancer patients or non-cancer individuals over lifetime. We all know that our cognitive abilities decline with aging. This is a normal process, but perhaps this decline is amplified in cancer individuals who have survived the disease, but who have had a history of a lot of treatment that could be harmful to the brain. Not everybody is affected, but this is a concept that has been evolving over time, and there are now a lot of task force at the NIH and FDA to study this mechanism much more closer of how aging can be influenced in the context of cancer treatment. So to summarize, some of the key points I have made so far is that there are certain factors that really are harmful to the brain, especially if they're all applied at the same time. Chemo drugs can be, not all of them, but some of them are offenders to brain function. Radiation to the brain can be a big issue. Oxidative stress, inflammation or chronic inflammation, aging per se, and that can be really amplifying the onset of neurodegenerative diseases. But genetic factors play a role, as we know, since some individuals are much more affected than others. On the flip side, there are factors that are actually positive and that have been studied, and that will bring me over to the next segment in a moment on management, which is exercise. Physical cardiovascular exercise in particular is such a powerful mechanism of really helping to overcome harm to the brain and to form new nerve cells. There's a lot of literature in high-profile journals to support that cardiovascular exercise is helping to form new nerve cells throughout life. The concept of enriched environment is very, very closely related to exercise. What is enriched environment? That just means that in a modeling system, for instance, in a mouse cage, where you have mice exposed to toys and tools and tunnels and things that can explore, they do so much better than a mouse that has nothing surrounding itself, just the cage and food. And so if you apply this concept to humans, you will immediately realize that an enriched environment, such as learning different languages, traveling, which unfortunately we cannot do a lot right now, but learning new skills. You've heard about the joggling or navigating through London. So using different modalities that your brain can do and really incorporating them through music and exposure to colors and smells is all adding to an enriched environment, which is very positive to overcome harm to the brain. I have not talked much about caloric restriction, but the concept of how diet and eating and overeating could be perhaps harmful by driving oxidative stress or eliminating oxidative stress is a very important concept that we just are not well educated in. But it is scientifically, at least, an important concept that influences the way we age and influences the way we are exposed and manifest certain side effects from cancer treatment. And antioxidants I talked a little bit about, and then neurogenic factors. So that is all science, and that you will now hear a little bit in the last segment of my talk. So management and interventions. One of them is cognitive rehabilitation. That is certainly the old school and most established way of helping individuals that are affected by cognitive dysfunction. With that in mind, usually the first step is to obtain a thorough neurocognitive evaluation, usually done by a psychologist or by a neuropsychologist, by really assessing all the domains that can be affected, doing attention tests, processing speed tests, assessing for memory function, for how you retain information. All of these are important factors that are relevant. And with that, then to identify goals of where patients may need assistance or where they may need help to overcome certain deficits. And that can include, as you can see here, compensatory mechanisms, strategies. So if you don't remember well, how and what can you do at least to come to an answer of having perhaps a notebook somewhere, notepads, alert systems in your smartphones that will remind you on certain things you should not forget. Going back to the exercise paradigm, so this has been now studied not only in animals, but also in humans, and is perhaps, in my eyes, one of the strongest drivers, more than anything else, more than cognitive memory games and more than medications can offer, is exercise and enriched environment as the biggest driving factor for longevity and for maintaining youthfulness throughout as we age, and to compensate for damage that occurs on the level of our brain, not only from chemotherapy, but from any other offending insult. And so it's never too late to start running. And here's an example of Mr. Singh, who started his first marathon at age 89. It's just an example of this sort of factor of exercise, cardiovascular exercise in particular, is working at any age. And as long as you can maintain it with maybe daily walking or building this in, like your usual routines that you do and have breakfast and brushing your teeth, going also for a walk or do some cardiovascular exercise on a stationary bike, these are very powerful interventions that you can adopt and build into your daily life. This is a very complicated slide, so don't dive too much into the details. What I wanted to bring away or take away from this slide is that aging is intimately connected to caloric restriction, oxidative damage, mitochondrial function. All of these issues that also play a role in the context of chemobrain. And that's something that deserves much more attention in the future, how certain nutrients or dietary changes could actually help us to have better quality of life in the context of chemobrain. Here are some of the drugs that have been used successfully in preclinical studies. So N-acetylcysteine, NAC, is an antioxidant that has been used to compensate for chemotherapy-related damage. Fluoxetine is actually an antidepressant that acts on the serotonin, which is a key amino acid pathway, which is critically relevant for forming new nerve cells. And then others you might have heard from donepezil, for instance, in patients that have been diagnosed with Alzheimer's disease. They may take this drug. This also has been used in patients with chemobrain. And then the issue of exercise that you just learned about. So these have been studied in animal models, and what can we do to really bring a lot of that knowledge to the bedside, to patients, and to make really increase their performance and improve their quality of life as a cancer patient and as a cancer survivor. So radiation is an easy one. Radiation to the brain. I haven't talked much about radiation, but it's a big offender to cloudiness and memory impairment. And so avoiding certain areas in the brain, such as the hippocampus, while you might need radiation to the entire brain, but sparing eloquent areas that are necessary for memory, for instance, is one of the ways that we now, in practice, treat patients. So hippocampal sparing is the key word. It's been done at a lot of facilities across the United States and worldwide. We could reduce the dose. Maybe you don't need such a high dose of radiation if there are efforts made to modulate this. maybe different modalities, protons versus photons. So different types of radiation could be considered, such as in children who are still in development, have not really developed or fully mature their brain function. And so if you treat kids at age less than 7 years of age or less than 5, they are much, much higher at risk for permanent neurological damage. And so thinking creatively about ways how we can defer treatments or spare eloquent areas is very, very important. And here's just an example how that happens. You really block out the hippocampus, which is this part of the brain in both sides, while a patient undergoes radiation treatment. Some of the drugs you just learned about from preclinical studies, but antioxidants and anti-inflammatory drugs, some of them you've heard as someone who might be treated for diabetes, cytoprotective agents such as this drug that has been used in Alzheimer's, or memantine is another one that has been used in patients with dementia, or neurostimulants. Neurostimulants you may be familiar with as another term is Ritalin, methylphenidate. Ritalin is used for attention deficit disorders. So that has actually been used also in adults who have been affected by chemo brain, just to help them raise a little bit more their level of maintaining attention and concentration, and can be quite beneficial in a subset of patients. And so to give you a little bit of a positive outlook, there's a number of clinical trials being sponsored and active right now that are visible on the so-called clinicaltrial.gov page. And many of these drugs that you just heard about, the methylphenidate, the Epicil, memantine, but also other drugs that have been shown in cell models and preclinical models to be protective to the nervous system, are now being investigated in different indications: breast cancer patients, in patients with head and neck cancer, lung cancer, and even any cancer, basket trials. And so how has that informed us really to bring that or form that bridge between basic science and patient management, and what are the mechanisms that play a role? So exercise, you heard about neurogenesis, the way of new nerve cells being born and formed throughout life, how that can interfere and slow down aging and increases brain plasticity. We talked a little bit about dietary interventions that might be relevant in individual patients, sleep modulation, avoiding toxicity, certain factors which we have learned now, genetic factors that we cannot perhaps influence, but at least allow us to identify patients at much higher risk than others. And then aging itself as an independent mechanism has given us a lot of clues to understand why brain and chemotherapy-related damage occurs, and what are similar mechanisms that play a role in both of these conditions. So showing you the same slide again, I think the holy grail right now in a lot of efforts, including our own research team at Mass General, is to restore this neurogenic niche in the brain. What can we do to make this cellular unit system in this area called the subventricular zone or the hippocampus? How can we make this area more robust, less vulnerable to treatments, to environmental toxins, to chemotherapy? And how can we allow this particular cellular niche to form better, to even form new nerve cells, and to help stimulate this particular mechanism? And I give you one exciting news, which is a paper that we have published just recently, that something that may affect you, because many of you have been treated for bone marrow related disorders, is how the bone marrow is actually talking on a daily basis to our brain. And you may say, this doesn't make any sense. Why would a distant organ system have an impact how the brain works? The truth is that in the context of injury, we have found out the surprising link that bone marrow derived cells circulate to the brain, where they can engage in repair processes. And that was done using labeling of cell types in green, where we could track them, where exactly they go, what they become, how long they stay. and study them in the context also in brain function, in learning and memory. And so this is a concept that is very, very exciting because it could be explored for the better and maybe to offer even a strategy to improve brain function over time rather than just using things like a Band-Aid to say, well, here's a memory aid for you, we'll help you to compensate for issues, but what we really would like to do is actually improve brain function after sustained injury. And so here's just the cartoon in a very simple version is you have the neurons that act in concert with these myelin-forming cells. They're happily together, they need each other to maintain brain function. And NPG stands for neural precursor cells, so it's a cell that is there throughout life that forms these adult new cells on an ongoing basis. And this is the normal situation. Now in the context of injury to the brain, such as radiation, these cells really get knocked out. So you have a loss of these progenitor cells. They don't form as many neurons anymore. Oligodendrocytes, the myelin cells suffer and fall apart, and brain function is just not optimal. But in the context of mobilizing some of the bone marrow derived cells and sending them where they will find their way on their own to the brain, they engage in stimulating this self-renewal of these progenitor cells, and as a consequence, make way more nerve cells that normally would be there without the input or influx from bone marrow cells. So it's a fascinating concept that has not been really explored, and we are planning right now a clinical study to help advance this system and perhaps study whether this also holds true in patients, in humans, as this was, to keep in mind, an animal study and not done in patients. So just to remind us, it's not just one thing. There are many different factors that are necessary for us to maintain nervous system function and to achieve our goals. And many different cell types and blood vessels need to act in concert to really maintain the complex function of the nervous system. And the cognitive function is just one of them. The way we think and the way we remember and the way we can execute certain tasks. And so the last slide is a little bit just to show you how we think the field can advance for the better of the management of our patients, which is using some of these preclinical model systems to apply them to develop ways and strategies to provide neuroprotection in patients. And that goes through screening of certain drugs, which ones are the most detrimental to brain function, and at the same time developing repair strategies, and study this in ways such as imaging or with neurocognitive assessment, whether there's actually a functional improvement with a neuroprotective strategy, and then obviously apply this to the patient scenario, to the human system through neurocognitive assessment. How can we maintain neurological function using advanced imaging modalities to see whether indeed brain structure changes over time? There are no biomarkers to be developed that will allow us to distinguish patients at highest risk and at lower risk, and eventually study protective mechanisms in clinical trials to get conclusive answers and evidence whether there is a drug that deserves more attention than others to get advanced and perhaps be one day FDA approved. So with that, there's a long list of many of my collaborators. This is obviously work that I try to give you a brief snapshot, but we've worked on these issues for approximately 15 years, have received support from federal sources and foundations, and many colleagues across the United States and also in Europe have contributed to some of the work that I presented today. So with that in mind, I would like to thank you for your attention, and I'm very happy to answer any questions you might have. And I think I will, at this point, stop sharing my slide and give it back to our moderators.
Thank you so much for the wonderful presentation. We do have a few questions. Our first question comes from a patient whose most recent chemo ended in 2016. They'd like to know, can I still have chemo brain, and how long does it last?
That is a wonderful question, and I appreciate that comment because I really didn't touch upon this issue at all in my presentation. The truth is that from all the large studies we know, and the most robust data is from patients obtained who had been treated for breast cancer, is that there are various phases that may be relevant for displaying symptoms of chemo brain. So while a patient might typically show symptoms in the first year, some patients show significant recovery and may not really have much in terms of symptoms after 4, 5, 6 years, whereas a subset of patients might actually have a worsening initially, but then they reach a plateau from which they really don't show a significant recovery. So really stay at a certain level that might be somewhat below their normal functioning that they had years prior before they were diagnosed with the disease. And there's some data that even looked at 20-year outcomes and found that a subset of patients does indeed have persistent deficits, but that is not the majority of patients. So the good news out of this is there is evidence of recovery that can occur over time. This recovery typically happens within the first 1, 2, or 3 years. So maybe at an age, at a time interval of 4 to 5 years, this might be more in a scenario of a steady state. But with some of the intervention that I outlined, there might be still some room for improvement and also to help you, whoever asked the question, to compensate and to feel better over time.
Thank you. Our next question comes from a patient who has not received chemotherapy, but appears to be having brain fog from very severe aplastic anemia. The question is, can you have brain fog from not having chemotherapy that's more related to a specific disease?
Yes, I think this touches on an area that has been very poorly investigated. But from the last few slides of my talk, you may guess my answer, which is the brain is connected to the bone marrow, to the function of the blood system. And in patients who might have profound damage to the blood system, may it be from chemo drugs or from radiation or even from the disease itself, it is not surprising to develop a condition that mimics some of the features that you have heard that we see in patients with chemo brain. This is not, as I said, well studied and certainly deserves attention of how and to what degree patients with profound impairment of their bone marrow function could also have complicated memory function or complicated cognitive function. So I would say, in short, I think there is a very likely chance that this is true, what you experience, but it's not been well studied, and I can unfortunately not give you any specific numbers of how often this is and to what degree and how this compares specifically to the condition of chemo brain.
Thank you. Is chemo brain more associated with long-term or short-term memory?
That's another great question. I think it is really mostly short-term memory, and the two different forms of short-term memory is the working memory and the remote memory. So the working memory is really, if I would ask you now to do a test that we do in clinical practice, which is serial sevens. You take 7 away from 100, how much do you get? 7 away from that number and 7 away from that number, and just try. It's not really a math test, it's how can you be attentive and also remembering exactly what you're doing, or I'll give you a number with 6 or 7 digits, and you have to remember this number and just give it back to me. So this is the immediate memory that can be affected. And the remote memory is your memory that is relevant if I tell you a story now, something that happened on the news, and then maybe in an hour or two, you really don't remember exactly what I said, or I asked you to remember a few names, and then we'll test you again in 10 minutes or in an hour, whether you can remember these names and give them back to me. This is what is mostly affected. It's not the long-term or chronic memory. For instance, things that happened years ago, things that happened to your childhood. See, there's a very, very resilient to damage. So I think much higher risk of having difficulties with a short-term component of memory and not with a long-term component.
Thank you. What are the therapies associated for issues with long-term memory? And that goes with the, that's the second part of the question that was just asked, is chemo brain more associated with long-term or short-term memory?
Yeah, so I think it's not associated really with, as I said, with long-term memory difficulties. If that is the case, one may have to consider other etiologies that are unrelated to cancer or unrelated to the treatment, such as a primary neurodegenerative condition where long-term memory could suffer as well. But it's not a classic picture that we do see in patients who are affected by chemo brain.
Thank you. Have the treatments for aplastic anemia, such as ATG, cyclosporine, and prednisone, been studied for chemo brain? This is coming from a patient who was treated in their 20s and then again in their 40s once they relapsed.
Yeah, another excellent question. I think the short answer is no. The long answer is some aspects of it might have been tested, but not in the context of aplastic anemia and the specific drugs that are used. I can tell you, however, that steroids, which is prednisone is one of the steroids, has been very well studied and can interfere with issues of sleep and can interfere with issues of cognitive function. So chronic exposure to certain steroids can indeed be a caveat that needs to be considered. But drugs like cyclosporine, for instance, I'm not fully aware of studies that have definitively assessed the impact on brain function. But it's worth investigating, I think.
Thank you. Does chemo brain affect your chances of getting Alzheimer's or dementia?
I would say the short answer is no, because there is no clear link that one is directly related to the other. But assuming you are a patient or someone is a patient who has a risk, a high family risk, for instance, to develop Alzheimer's disease, and this Alzheimer's disease manifestation would hypothetically show up at age, let's say, 70. And you are now treated as a 60-year-old for cancer, it is possible that the manifestation of the neurodegenerative conditions, such as Alzheimer's disease, would occur somewhat earlier than it normally would have in the context without any cancer or cancer-related treatment. So this goes back to this concept of accelerated aging, that we think certain drugs can interfere and accelerate that process. But I think if you have no risk for Alzheimer's, you will not get Alzheimer's disease or a similar condition from chemotherapy drugs. That is unusual and not really expected.
Thank you. This question comes from a patient who says they've noticed severe loss of recall ability after ATG treatment. It seems to be chemo brain. Is this a possible side effect? They've noticed a greater problem when their hemoglobin level counts are down.
Right. So there are actually two questions buried in this question, which is an important one. The first one is actually, we touched upon this earlier, whether some of the drugs that are used for aplastic anemia could interfere with this. And my answer was, I cannot rule this out. This has not been well studied. And we also have not specifically looked at this drug and its impact on brain function. However, what I can say is that anemia itself can be a problem, and a lower count of red cells that can drive fatigue can be a major influencing factor for brain fog. And that's why drugs like erythropoietin that can drive rejuvenation of the red blood cell system have been used to improve issues such as fatigue, or other drugs have been used to compensate for fatigue-related mechanisms. So I think it makes perfect sense if the observation is that with a lower red cell count, things are worse. That is not surprising and has been observed with many other conditions as well. So fatigue is a major component, and obviously efforts should be made to improve level of fatigue in order to enhance cognitive function.
Thank you. If you have attention deficit disorder before chemo, how would that affect you differently in regards to chemo brain than someone without ADD?
I don't know the answer specifically to this question, but I can speculate that I would say the risk of having issues with attention and concentration and processing speed and getting a task done is probably higher in someone who has such a pre-existing condition, but also may suggest that drugs used previously might now have a role again to be used to overcome some of these symptoms. But it's not an area that has been well studied, similar to the drugs that are used in aplastic anemia. That's something one should look at and really provide some more comprehensive answers to see if someone who has ADHD as a child, for instance, whether they have a higher risk to display symptoms of chemo brain later on in life if treated for cancer.
Thank you. Our next question is for folks who have had a bone marrow transplant. Do they have any amount of protection from chemo brain by having added progenitor cells circulating through the body after transplant and until implementation of the bone marrow?
That also is not entirely clear. What is known, however, is that patients who do have a bone marrow transplant are at higher risk for developing cognitive and mood-related issues. And that really goes back to the main axis that bone marrow is relevant for brain function. The good news, however, is if the bone marrow function can be fully established over time with all the supportive care, is that there is no reason to believe that there is not also room for improvement. So I am not able to specifically answer what sort of the time window is and what exactly happens, because this is a very novel field of investigation. But there is literature emerging that patients who've had bone marrow transplant, such in the context of lymphoma or leukemias, are at higher risk for developing cognitive issues.
Thank you. Does chemo brain or brain fog seem to affect men more frequently or women, or it doesn't matter?
I wish to have the answer to this, but it is not known. I think what is known that breast cancer patients have been the most vocal about it. And this is the disease group that has been best studied with really abundance of literature. I would estimate about 75% of the literature we have in this field has come from breast cancer survivors. And so the majority of them are obviously female. And men tend not to talk about these issues or raise it necessarily as often when talking to their doctors. So I would say that there is maybe some bias built within, but I would speculate that it actually affects both genders equally or somewhat equally but we don't have good data to really have a comparative analysis longitudinally. And so when I mean longitudinally means you have the ability to have a baseline before you get even treated. And then we will look again in terms of cognitive function through cognitive tests in 5 years and in 10 years. And on an individual level, know what the difference is. Because one thing I also didn't make clear in my presentation is that if you are someone who has been living on a very high cognitive level, let's say you're a multitasking manager type, or even someone who stays at home and takes care of 5 kids and runs a business, and you do multitasking a lot, for you having a decrease in capacity by 5 or 10% may be a major challenge. And others around you might not even notice that. They could say, listen, do you completely normal? Everything is, no, I'm not. I just could do these things with ease and I just can't anymore. So there is, on an individual level, a huge subjective threshold that is notable, and it needs to be exonerated, and we cannot just simply brush over on a cohort number, meaning I assess, let's say, 100 subjects and all 100 do reasonably well after a year or two, if a few individuals suffer a lot and notice that. So what I'm saying with this is that sometimes if you perceive symptoms, they might really be quite profound on your individual level and not necessarily notable if compared to a large population.
Having chemo brain or brain fog can be very sometimes difficult for families and friends to understand that the patient may be suffering from this. Is there some best advice on how patients can let others know what they're experiencing in a simple way? Because it can be very complicated to explain what you're going through as a patient. Is there some best advice on how to explain what you're experiencing so other people will understand what you're going through?
Yeah, I think there are some good websites. One is from the American Cancer Society that explain what chemo brain is and what the symptoms are, similar to what I tried to outline today. And I think in the most simple terms, if someone would ask me, what is chemo brain? It's just essentially a condition that doesn't allow you to use your brain as its best ability. And that, in turn, means you don't remember things as well, and you cannot concentrate on a task and be as fully attentive. So family members might perhaps understand this, that having gone through cancer and intense cancer treatment will take a toll on subtle functions such as cognitive abilities. Even so, everything else seems to be normal and the motor function is good and you're able to get around and get things done, but maybe things just take a little bit longer to get it accomplished, and maybe sometimes even forgotten, where you need memory aids. So this is a condition, as I said earlier, that is very common, and even so can be mild and can be reversible in some individuals. Others could suffer for years to some degree.
And our last question is there an average time that you see patients regenerate cognitive function post-treatment? This patient has felt foggier or slower for the last 5 years since a chemo treatment, and they're curious, is there a normal regeneration period to feel normal again, or is it truly circumstantial for each individual?
In short, I think it is circumstantial for each individual. It's very unique. There is no classic evolution that everybody follows exactly the same path. But from my experience of having seen so many patients with cognitive issues in the context of cancer therapy, I would say the majority of improvement will happen in the first 2 years. And I do have patients, however, who feel better in year 3, 4, and 5. On the flip side, if I see someone who had been treated 8 years ago and they have ongoing issues, I think the chances are less hopeful that there will be a significant improvement in years to come. So I would say, in very simplistic way, the biggest improvement typically is seen in the first 1 to 2 years, but can be up to 5 years. And the chances are smaller beyond 5 years to see significant improvement.
Well, thank you so much, Dr. Dietrich, for spending your time and your expertise with all of us today and sharing this wonderful information to help us all better try to understand chemo brain and how it affects patients. If you would like to watch this webinar again at a later time, it will be available on our website within 2 to 3 business days. If you have any additional questions, you can send it to us at any time in 3 ways. Send an email to help@aamds.org, call our helpline at 800-747-2820, extension 2, or submit your question on our Facebook page. On behalf of the Aplastic Anemia and MDS International Foundation, thank you for joining us today and making us your resource of choice for information on bone marrow failure diseases. The AAMDS-IF Medical Advisory Board and team are here to help you and your family as we have done for the past 36 years. This concludes our program. Thank you, Dr. Dietrich.
Thank you very much.